So, dear audience, my name is Anna Grassalkovich. I'm a fifth-year medical student, and my mentor is Professor Peter Hegyi. And our research group is mainly focusing on the treatment of acute pancreatitis. Acute pancreatitis is one of the most frequent gastrointestinal diseases that needs acute treatment in hospital. The main factors of this inflammatory disease is alcohol and some biliary diseases such as bile stones. The mortality in severe cases can reach a very high percentage. The reason behind this high mortality that there's no specific treatment available yet, which urges the identification of new drug targets. During my research, I'm mainly focusing on the effect of alcohol. But to do that, we had to understand the physiology of the exocrine pancreas. Pancreatic acinar cells produces more than 300 bioactive molecules and many hydrogen ions. Therefore, the pH is very low near the acinar cells. In acidic environment, the autoactivation of the trypsinogen can happen easily, which can lead to pancreatic inflammation such as acute pancreatitis. Therefore, the function of the ductal epithelial cells is essential, since they produce bicarbonate-rich alkaline fluid that washes out the digestive enzymes. In this process, the CFTR channel, which is a chloride and bicarbonate channel, which is expressed on the apical membrane of the ductal epithelial cells, has a leading role. If this physiological function of the pancreas damages, that can lead to pancreatic diseases. It is known that CFTR gene mutations in cystic fibrosis patients, which is one of the most common genetic disorders, can lead to pancreatitis. But it was not known that etiological factors that causing pancreatitis can also damage the CFTR channel. And our research group firstly showed that alcohol can damage the function and the expression of the CFTR chloride channel. So therefore, we already have a clinically approved medicine called Orkambi, which is both contains an activator and a corrector. So there's the question, if the etiological factors that causing pancreatitis damage the CFTR channel, if we use this medicine, it can be a good method to treat acute pancreatitis. This is a translational question, which means that we had to go from the basic sciences till the clinical trials. During my research project, I'm mainly focusing on the basic and applied sciences. These compounds of the medicine called Orkambi are affecting two pathways. VX770 increases the open state probability of the CFTR channel, whereas VX809 correcting the damaged CFTR expression. So our aim was to test the clinically approved medicine, Orkambi, in alcohol-induced pancreatitis. We used guinea pig pancreatic ducts with enzymatic digestion, and we treated these ducts and used immunofluorescence staining. The immunofluorescence staining mainly mimics the binding of an antibody to an antigen. After that, we used confocal microscopy. In all of our pictures, the green one is the CFTR channel, and the blue ones are the nucleus. At first, we would like to map the effect of alcohol on the CFTR expression. On the control ones, you can see a nice CFTR staining in green on the apical membrane of the ductal epithelial cells, which means that these pancreatic ductal cells are polarized cells. They have an apical and a basolateral membrane, and the CFTR is always expressed on the apical membrane. And due to the alcohol, the expression of this channel is almost completely destroyed, and also the histological structure of the pancreatic ducts is damaged. We quantified our results, and you can see the decrease of the fluorescent intensity due to the damage of ethanol. After that, we would like to test the CFTR potentiator and the corrector alone if they have any effect on the CFTR expression. And we found that they did not have such a big effect on the CFTR expression. After that, we added the corrector to our system. And you can see that in normal conditions, there is a big peak in the intensity of the CFTR channel because that apical membrane, you have the CFTR expression. This is completely destroyed due to ethanol, and if we add the corrector to our system, it's restoring. And also we quantified our results, and you can see the restoration of the CFTR channel in these sheets. After that, we would like to map the exposure time of CFTR of the corrector. This is an important pharmacological thing to see, and we found that even 2 hours of exposure time was enough for this corrector to make the effect. We also would like to map the dose dependency of this corrector. These compounds are very expensive. For 1 year, it's 80 million forints to treat just one patient. So it's really important to find out that which concentrations are good to use. And we found that 5 and 10 micromoles of the corrector is very efficient. We did the same experiment with the CFTR activator, and we also found out that the 5 and 10 micromole of concentrations were the best way to treat our ductal cells. After that, we would like to test the combinatory effect of this medicine, because in the medicine, I said that they are in a combinatory treatment, and we used the CFTR corrector and activator alone, and then after in combination. You can see our results that even the CFTR activator had correcting effects. You can see that in the purple sheet. And the combinatory treatment was very efficient, while the 2 compounds did not potentiate each other's effect. After that, we also tested the combinatory treatment in different concentrations, and we found out that also the 5 and 10 micromoles were the efficient ones. And after, we evaluated the concentrations of ethanol, which can make severe and a mild alcohol intoxication. And the protective effect was at 30 and 50 millimoles of ethanol concentration, which can make severe and mild alcohol intoxication. With 100 millimole of ethanol, the protective effect is decreased, but that concentration is almost deadly to patients during alcohol intoxication. Of course, we have some more further questions to answer. If ORCAM-B, besides the correcting effect, will also have the functional corrector effect, and we would like to test the compounds of ORCAM-B in experimental pancreatitis models. So now we are currently working on a guinea pig pancreatitis model, and if they are successful, we can move to phase 2 clinical trials, which means that we could test these compounds on acute pancreatitis patients, because the phase 1, which is the toxicity trials, were already finished during the clinical approval of ORCAM-B. I would like to thank my co-workers for their help and you for your kind attention.